Hello, my name is Shingo. I'm a senior scientist at Okinawa Institute of Science and Technology, Japan. Let me introduce myself first before the talk. So I did a PhD in fungal biology in Australia. Then I moved to bioinformatics in my early postdoc. Then in my later postdoc in France, I started developing visual tools or bioinformatic tools to deal with the huge amount of genomic, transcriptomic, and proteomic data. And after 7 years of trials and errors, we have established a set of visual tools for fungal omics. So today I'm going to talk about a visual integrative omics of fungi. This is the outline of my talk. I'm going to quickly cover the background information on JDA fungal genomes and our visual tools. Then I'll talk about a set of established visual tools. We call it Visual Integrative Omics Platform. Then I'll give you a visual example using the visual tools, the evolutionary gene loss in ectomycorrhizal fungi. That leads to a summary of my talk. History of mycorrhizal genome sequencing. Joint Genome Institute, Microcosm, hosts over 2,000 fungal genomes under the 1,000 Fungal Genomes project. Over a decade ago, Mycorrhizal Genomics Initiative was launched under the 1,000 Fungal Genomes project. Around that time, the first mycorrhizal fungal genome was published. In 2015, comparative genomics of 30 mycorrhizal genomes was published. In 2020, the sequel comparative study of 62 mycorrhizal fungi was published. Today, there are over 200 mycorrhizal genomes available on Microcosm, including ectomycorrhizal, arbuscular, ericoid, and orchid mycorrhizal species. Okay, let me talk about the background of my research projects. I've been involved in about 20 fungal genomics projects, including wood decayers, ectomycorrhizae, arbuscular mycorrhizae, and endophytes. There is always great demand for digesting fungal genomics information. And our aim was to simplify complex omics data in such a way that results can be linked to biological context. So we set out to explore data visualization, and we have developed a set of visual tools. We call it Visual Integrative Omics Platform. Let me explain the concept of our visual tools. We can combine various omics data and visualize results for easy biological interpretation. We can grasp a genome-wide view of biological activities. Then, once we spot interesting patterns, we can zoom in and examine further. The approach is a bit like a Superman watching the entire population on Earth, instead of watching one person in a village. What I'm trying to get is we want a genome-wide view instead of going through gene by gene. Here are some examples of visual work in our recent papers. Our visual omics analyses have contributed to international collaborative studies. We have published 21 papers with 541 citations in the last 7 years. So let's talk about the Visual Integrative Omics Platform. So we have developed innovative visual tools. First one, co-expression analysis for combined genomes, transcriptomes, and secretomes with machine learning. The tool is called SIM+Go. We can compare repeat elements in genomes with a tool, Tingo. We can compare genomic features with a tool, Pringo. We can compare synteny with a tool, Singo. And we can integrate any of these above and visualize them all in a single plot using Vingo. So we can tackle research questions by combining the visual tools. Okay, let's move on to a visual example, the evolutionary gene loss. A signature of the genome evolution in ectomycorrhizal fungi is the loss of genes for enzymes degrading plant cell wall polysaccharides. Let's have a look at the figure here. It shows the copy number of genes for plant cell wall degrading enzymes in a variety of fungi. There is a clear reduction trend from white rot fungi on the left top to ectomycorrhizal fungi on the right bottom. Here is the question. Many plant cell wall degrading enzymes have been lost in ectomycorrhizal fungi, but what exactly happened to the gene lost in the genomes? So we investigated the evolutionary gene loss of CBM1GH6 silobiohydrolase in volitalis. It was published in 2021. So the figure shows the count of GH6 module. There are ectomycorrhizal and brown rot species in volitalis. Now, GH6 is present in brown rot species, such as Hydromerilius pinastri, the species in the middle of the figure. But the GH6 is absent among the ectomycorrhizal species. So we selected several fungi and visually examined syntenic regions containing CBM1 GH6. The idea here is that in evolutionary conserved genomic regions, also known as synteny, similar genes tend to be located among closely related species. We find such regions containing CBM1GH6 in H. pinastri and the related ectomycorrhizal fungi. Then we trace a remnant of gene sequences for CBM1GH6 in the related ectomycorrhizal fungi. To do this, we identified synteny among the selected species with single pipeline, merged with the genomic locations of cousins, and finally visualized the entire genomic scaffolds of the species using Vingo pipeline. The figure shows evolutionary conserved regions for cousins. The species are in the evolutionary order. The density of all genes is shown in gray upward peaks. The density of repeat elements is shown as a gray downward peaks. The syntenic regions between the species are linked with blue lines. Here is a conserved region containing cousins and CBM1GH6 silobiohydrolase in brown rot Hydromerilius pinastri. A set of cousins in red, GT22, AA51, GT58, and GT1 is conserved among the fungi compared. The corresponding location of missing CBM1GH6 gene in ectomycorrhizal fungi is given as a green lollipop. So we visually identified the remnant of CBM1GH6 in the ectomycorrhizal species at the scaffold level. In summary, with our visual tools, we can create a genome-wide view and visually pinpoint the gene decay of CBM1GH6 among the ectomycorrhizal fungi. The visual approach enables us to see global and local patterns in complex genomic data. We can compress large data into one figure, and such a figure gives a lot more impact than a long monotonous paragraph. If you were interested, please visit our website on Visual Integrative Omics platform. I'd like to thank all my colleagues and the collaborators involved in the fungal genomics projects. There are so many people involved in, and I cannot fit everyone in this slide. I am so sorry. Here I put my main collaborators around the world. Unfortunately, I don't have time to thank people individually due to the time restriction, but I have many collaborators in France and Germany, where I did my postdocs in the last 7 years. And there is one last thing. Can you see a red mark saying OIST on the edge of the world map? And that's where I currently work. This is a subtropical island called Okinawa. I recently moved there. We are currently setting up a new laboratory for both bench and computational work with the newly appointed Professor Fyodor Kondrashov, Evolution and Synthetic Biology Unit, Okinawa Institute of Science and Technology, Okinawa, Japan. If you are interested in doing PhD or postdoc, please contact us. Also, I am personally looking for collaborators to explore terrestrial and marine fungi. So if you have a unique fungi to share and willing to sequence genomes, we can work together and make it happen. I am very casual and friendly, so please feel free to contact me. So, this is the end of my talk. I hope you enjoyed it. Thank you very much indeed for your attention.